Are going to jump into all of the fun science presentations. Yay! For all of our awesome therapies that we have. So exciting. And we're going to start out first with Bill Baker from Denali Therapeutics, and we'll let you take it away for the DNL 310 study.
Thank you. Yes, I get to kick off the fun science part. So thank you so much for having us. Thank you so much for having me. And I have to say, I don't often have the opportunity to come to meetings like this. And so it's nice to be reminded of how powerful a community can be for all of us. So thank you so much. Oh, there we go. Okay, so for my disclosures, my name is Bill Bacher. I'm a medical science liaison with Denali Therapeutics. So obviously my disclosure is I'm employed by Denali. But also I'll be discussing DNL 310, which I may alternately refer to as TV Denafusp alpha. You're not obligated to remember right now. But that is an investigational agent and it's not currently approved by any health authority yet. So for an overview, I'll just talk about Denali and who we are for those who in the room who are not familiar with us. And then I will go into some of the information on our MPS2 development program, as well as DNL 310. So who are we at Denali? Well, our purpose is to defeat degeneration. It's been our very singular laser-like focus since 2015 when the company was created. You can see in the lower left of the slide some of the disease areas that we focus on, particularly with neurodegeneration like Alzheimer's and Parkinson's. But importantly for us all in this room, and definitely importantly for me as well, is our focus on lysosomal storage disorders. And really what we've done since our inception is we've had a very strong commitment and understanding of the power of community and how listening to patients and families and listening to the community is so important, because at the end of the day, all of this is for you. And so you have to have a voice in all of this. So thank you. As far as our development program for MPS2, we have completed a phase 1/2. That's a small phase 1/2 study with DNL 310. We also have currently enrolling a larger phase 2/3 trial that's called COMPASS. There are sites in South America and Europe and North America, even very local to where we are sitting right now. And then finally, we announced last month that we are pursuing the filing of a biologic license application with the Food and Drug Administration under the accelerated approval pathway. So what is DNL 310 for those that are not familiar? It's called a fusion protein, which means that there's two proteins that are bound together. The first is the enzyme that's relevant for MPS2, of course. And the second is what's been developed at Denali called an enzyme transport vehicle. What this portion of the molecule does is it attaches to a receptor on cells called the transferrin receptor. This receptor is everywhere. But importantly, it's on the cells that create the blood-brain barrier. So when DNL 310 attaches to the transferrin receptor, that transferrin receptor moves across the blood-brain barrier, making DNL 310 available to treat the symptoms of MPS2 inside the central nervous system. But also because that receptor is everywhere, DNL 310 is available to treat the rest of the body as well. Now, regarding our development or our phase 1/2 clinical trial, I'll share some of the results with you right now. The trial enrolled 47 participants with MPS2. They were all given DNL 310 in an open label manner, which means the patients and the family and the investigator, everybody knew that DNL 310 was being infused. That was infused weekly. And the trial itself is 24 weeks, but we have some open label extension or some longer term data as well that I'll share. Regarding the safety, because safety is the primary focus of this trial, of the 47 patients, almost all of them had the neuronopathic form of MPS2. One participant did discontinue due to treatment emergent adverse events, in part of which was related to infusion-related reactions. 72% of participants reported that their adverse events were mild to moderate, and the most common adverse event is infusion-related reactions, which did decrease in severity and frequency over the length of the trial. The next couple slides are on some of the outcomes regarding biomarkers or things that you can look at inside of the patient in order to see what the drug effects might be. So this first one is heparan sulfate, which I know we all know how important heparan sulfate is to discuss. So this is heparan sulfate inside with the cerebrospinal fluid, which is the area that's around the brain. And what you can see from the slide here is that from the beginning of the trial at baseline week 0 to week 24, there was a 90% reduction in heparan sulfate levels in the cerebrospinal fluid. And then that does continue outward as the trial goes on. It's maintained as the trial goes on. Secondly, and similarly, are the heparan sulfate levels in the urine, which by week 24 also decreased just under 90%. And then those levels are maintained throughout a longer period of time as well. And then finally is neurofilament light chain. This might be something you're starting to hear a little bit more about. It's still an investigational biomarker, but the science is really emerging on neurofilament light chain and how it might be important. It's a protein that sits on neurons, on the nerve cells. So if the nerve cell is damaged, it can be released and you can look for it. So what we see in our trial is that while there's an initial increase from baseline to week 24, after week 24, on the average level of neurofilament light chain decreases quite significantly. You can see the slope of it and that it does then continue forward. So that wraps it up for me today. I just want to thank Project Alive for inviting us. Thank you all for being here, and a special thanks to the families and participants in our trials and the investigational sites who are working so hard for this. So thank you so much.
And just so you all know, we will have Q&A with everyone at the end. So if you have questions, mark those down and save them for the end. So next we are going to have our presentation. Are you here? Where's Robin? There's Robin. Okay. From JCR. So thank you so much. Let's welcome Robin.
Let's see. So thank you to Project Alive for hosting this meeting, and it's really just wonderful to see so many of you out here. My name is Robin Lewinter, and I am the JCR USA Medical Affairs and Medical Communications Lead, and I'm looking forward to sharing with you all a bit about who JCR is and our exciting platform designed to deliver enzyme across the blood-brain barrier and to provide new treatment options for patients with MPS II. This is our forward-looking statement. Leave it up for a second. So before I get into details about our platform and our therapy pipeline, let me introduce JCR, the company. So JCR is a Japanese pharmaceutical company founded almost 50 years ago now. And until recently, it's been predominantly a domestic company. The 49 years of its history we've focused on manufacturing, research, and development of process technologies and new treatment options, especially in orphan and rare diseases. As you can see, JCR is headquartered in Kobe, Japan, and where there's a number of other offices distributed throughout the rest of Japan, with 2 research centers focused on discovery and proof of concept of innovative technologies and platforms, and 5 manufacturing sites where we create the products that are marketed in Japan and our clinical trial products for our global studies. And I'd really just like to highlight our focus on development. More recently, JCR has expanded our global footprint in support of global clinical trials, and we have a presence in the USA and Brazil and the Netherlands to sustain our global clinical development and our programs for rare diseases. I'm now going to share with you the basic premise of J-Brain cargo technology and why we are so excited to explore its potential in lysosomal storage disorder. So as we all know in this room, MPS is a lysosomal storage disorder. And while initially it seemed a treatment would be simply giving patient the missing enzyme, we now know that the enzyme is not able to cross the blood-brain barrier. And as was just presented, the blood-brain barrier is a very selective filter that blocks the passage of substances into the brain. It's quite important because it blocks pathogens and things that you don't want in the brain to get into the brain. But because enzymes also unable to cross the blood-brain barrier, simple enzyme replacement therapy cannot treat the neuronopathic components of MPS. So JCR has created a technology called J-brain cargo that allows enzyme replacement that allows the enzyme to enter the brain by taking advantage of the body's carefully regulated blood-brain barrier. So how does this work? So on the left two kind of cells that you can see up there, all cells need iron to function. And iron is safely carried around the body in a protein called transferrin. And cells in the brain need iron. And in order to get iron into the brain, the transferrin has to be ferried across the blood-brain barrier. And our body has developed a mechanism to transport transferrin across the blood-brain barrier by binding to a specialized receptor, which is, cleverly called the transferrin receptor on one side of the brain, encapsulates the whole transferrin into the cell, buries it across the blood-brain barrier, and then releases the transferrin or the iron into the parenchyma where it's available for use by cells in the brain. So J-brain cargo has essentially hijacked this process, and you can see more on the right side, where we've developed a technique that fuses the enzyme to an antibody to the transferrin receptor. Now, if you remember, an antibody is kind of like a puzzle piece, and in this case, it connects to the transferrin receptor on the J-brain cargo, and just like with transferrin, the transferrin receptor encapsulates our product, ferries it across the cell and releases it into the parenchyma, where the enzyme is now available for use by cells in the brain. Now, we know that MPS affects multiple organs, both in the brain and in the body. So therefore, in addition to being able to cross the blood-brain barrier, it's important to note that as an IV infusion, this drug is available within the whole body to be used and can be taken in by both the receptor to the enzyme, but also the transferrin receptor, which was just described as on all cells in the body. So this is our, R&D pipeline. JCR is using this J-brain cargo to explore treating many lysosomal storage disorders, which we show in this slide. And for the rest of this talk, I'm going to focus on our lead program, JR141, or pabinafusp alpha. It's hard to say, so I will just call it JR141, for the treatment of MPS II. I'm now going to share out some data that led to the first approval of this drug in Japan, and it's only approved right now in Japan in 2021 for the treatment of MPS II. Approval was based on the reduction of heparan sulfate in the brain, which is, as we all know, is a biomarker indicative of neuronal involvement. So this is the multiple clinical trials have been done in patients with severe and attenuated MPS II using our drug using JR141. On the left, you can see the left shows heparan sulfate levels in the CSF, the cerebral spinal fluid of treated Japanese patient. You'll note that with an initial course of JR141, the heparan sulfate levels in purple there go down. Patients were then removed from the drug, and during that period of time, you can see a rebound in heparan sulfate levels. And then treatment resumed, and the heparan sulfate levels in the CSF again went down, and they stayed low throughout the rest of the treatment. On the right, you'll see the somatic effects of JR141, where here we're looking at spleen and liver volume. And you'll note that the somatic effects are equivalent to what you see with normal idursulfase or enzyme replacement therapy. So we are so proud that these studies have led to the development of an approval of Iscargo, which is the marketed product in Japan. It was approved in 2021, as I said, based on these clinical trials that have shown the reduction of heparan sulfate levels in the brain of treated patients. The safety profile of JR141 has been studied for over 8 years now and evaluated in 96 patients in clinical studies and 50 patients post-marketing. And all in all, there have been no significant findings, and the benefit risk is considered favorable. One of the benefits of us having a marketed drug is we are able to do post-marketing trials in patients who have been prescribed the drug and get data on real-world patient experience. So for example, here we show feedback from interviews with caregivers representing 7 Japanese children who received our drugs for around 3.5 years at the time of the interview. Most of these caregivers report a general trend towards improvement in multiple aspects, of their children's disease, including cognitive improvement, language, concentration, and self-control, all of these items that we were just shown that are assessed in the various behavioral tests. And there have been even some reports of improvements in the entire family's quality of life. We are now working to expand access of JR141 beyond Japan and are in the middle of a global phase 3 study designed to demonstrate the safety and efficacy of JR141 on the CNS and somatic signs and symptoms. It's called the Starlight Study, and more details can be found on clinicaltrials.gov or by using that code, or you can stop by our booth out front. The general study outline is here. JR141 is randomized against standard of care, and you'll note we're studying 2 cohorts of patients, a larger neuronopathic cohort and a smaller cohort of attenuated patients. The primary endpoint for cohort A, or the neuronopathic patients, is efficacy and safety at 2 years post-treatment. We are actively enrolling patients in this study at sites all over the world. Specifically for this audience, we have 7 sites open in the United States, in Oakland, Chicago, Minnesota, Philadelphia, New York, Phoenix, and 1 right here in North Carolina. So thank you for your attention. We are really excited to see the potential of JR141 in the treatment of MPS II.
Thank you, Robin. Okay, next we are going to welcome Ravi on the stage to tell us a little bit more about Takeda and your commitment to rare disease. So thank you. We had a little switch up in the schedule too, so you'll see that.
Well, a very good morning to everyone. It's truly an honor and a privilege to be here with all of you, and thank you, Project Alive, for inviting Takeda to talk to you today. Let me start on a personal note. 7 years back, I switched my career trajectory to start working in rare diseases. What I thought was a new job for me eventually turned into a purpose that has continued to guide everything that I have done. And I've had the privilege of working with a company that feels the same. I was very humbled right at the onset by the sheer amount of grit, determination, and love that all of you had for your children, for each other, and for the cause. And today what I wanted to do was give you a brief introduction of what we have been doing in Takeda over the years, where we are today, and how we would like to continue to be in this space. Some of you may be familiar with Takeda. It's a Japanese company. It's more than 250 years old. So we've been in this space for a very, very long time. And one of the core values of this company has always been patients first. And even today, that's something which we continue to hold on to. I don't need to walk this audience through the challenges of what rare diseases and their families face every day. But suffice to say that with more than 7,000 different rare diseases and less than 10% of treatments available, the unmet need is astounding. But at the same time, you also have to acknowledge that there is a long, painful, arduous diagnostic odyssey that families have to go through. It takes years and years of ricocheting around different specialities before finally getting to know what a true diagnosis is. And that continues to be a major challenge. 80% of these diseases are inherited, which means the value of genetic testing cannot be underscored. And then, even once you get through all this, barriers to care are something which very few people understand or even acknowledge. Geographic hurdles, access to center of excellences. UNC is a great example of how you can have the best world-class access to treatment, therapy, trials, but many of us don't live here. Many of us have to traverse that journey, and it's not easy. So at Takeda, we decided to do a couple of things to see how we could actually address some of these unmet needs. So we are proud to be a founding member of what we call the Global Commission to End the Diagnostic Odyssey for Children. And we last year put together a framework to accelerate diagnosis for children. And it's available online. The purpose of this framework was essentially to help both the providers, the families, and the advocates chart out a path which would hopefully lead to quicker diagnosis. One example is the Rust nomination of MPS2, in which I was fortunate to play a very small role, but I consider that one of the biggest accomplishments of my career so far, because that is what has allowed us to today ensure that children are diagnosed on day one and not 7 years or 6 years or 8 years or whatever that time frame looks like. The other thing that we decided to do was start working with NORD. This is the National Organization for Rare Disorders. You may be familiar with this. We basically wanted to focus on 4 things. Number one, increase equitable access to genomic sequencing. The second was to extend support for centralized and specialized care in diseases. Of course, reducing barriers to research participation, and also invest in accessibly, culturally relevant rare disease resources, because that's the other thing to keep in mind, that we have families from diverse backgrounds, with diverse needs, with diverse challenges. How can we make this more equitable? Because yes, you need treatments, you need access to treatments, but you also have to ensure that they are available for all. The other thing that Takeda did was we, through a medical grant, partnered with the Children's National Hospital of Rare Disease Institute in Washington, DC, on something called as the Rare Disease Clinical Activity Protocol, or RareCap program. So what this program essentially does is establish a network of development, dissemination, and curation of protocols that are aimed to standardize care. So remember, I just mentioned to you that not everybody has access to center of excellences. Not all physicians are experts in treating rare diseases. Where do they go when they have a question regarding a protocol? So previously, it would be emails, phone calls, and trying to reach out to their peers. But RareCap actually allows them to go online and look at protocols that experts share. So think of it like a wiki format where the community members, including patients, families, can put their feedback and the broader community has access to it. So that's something that Takeda is very proud to be associated with it. And with Dr. Marshall Sumner's, I think that would be a true game changer in the days to come. So what are our current initiatives? I'll tell you that we have based it on 3 main pillars: on evidence, education, and enablement. These are the 3 things which would drive not just what we do today, but what we'll continue to do in the future. So under evidence, of course, some of you are probably familiar with it. We have the Hunter Outcome Survey Registry, which is the single largest source of natural history, collecting data for more than 15 years. We also have different grants where we are focusing on biomarker identification. We are very close to publishing the readouts from the 100 Genome Project that some of you families may have participated in. It has been a fantastic learning experience for us, and Christian and other people are also part of that publication exercise. So we are looking forward to that. In terms of elevating knowledge via insights, we are continuing to invest heavily to generate real-world evidence in the form of a systematic literature review that was recently published. Basically, it will tell you everything that the current enzyme replacement therapy, which is approved, can and cannot do for you. And this has been curated through a rigorous scientific exercise. We continue to invest in continuing medical education, which trains and educates people about the disease area. And then, of course, enablement. I think this is the single most important thing, if you take away from my talk, is that Takeda continues to invest in this space. We continue to invest in newborn screening-based enabling studies, whether those are practical recommendations, which we are hoping to put out in the next few months, whether it is supporting Delphi consensus guidelines, which will help physicians have a more consensus-driven approach towards treatment, and of course, investing in the next generation of treaters and providers. So Takeda supports them through fellowships and training programs and so on. And I think that's how we would like to empower our care partners moving forward. So as I close, I would just like to close on a personal note. Remember, I told you I started my journey 7.5 years back. The first meeting of this kind that I attended, I happened to speak to a mother of an MPS II patient. And she told me that every morning I get up and do this, not because it's easy, but because my child is worth it. And I think that really stayed with me. So as I leave today, I want you to know that at Takeda, we are humbled, determined, and focused on standing with the community as you move forward through new technological innovations and making sure that life for you and the children is much, much better than what it is today. So thank you so much. Thank you for all your courage, grit, and determination. It's truly inspiring.
Hey, thank you so much, Ravi. And now we are pleased to welcome Nadal to the stage to talk about the RGX 121 study. So thank you so much.
Thank you. Good morning, everyone. It's my honor to be with you here today and to share REGENXBIO's story for RGX-121 treatment for MPS II patients. This is our forward-looking statement. I'm not going to spend time reading this. So what is gene therapy? Gene therapy has the potential to treat genetic diseases. And genetic diseases often have a defect in a gene, usually a single mutation. So what it does, it prevents the gene from having the correct instructions to make a protein that's important for the body. So through the therapy, we can deliver a functional copy of the gene using a very common terminology we call a vector. So you will often hear vectors when you are listening to gene therapy studies. And then one vector that was very commonly used is AAV. So AAV is a vector that is not known to cause diseases in humans. And there's a QR code here that if you have time to scan it, it will tell you a little bit more about the technology that REGENXBIO uses to deliver gene therapies. RGX-121 is a gene therapy that is specific to deliver a gene that replaces the IDS enzyme to the CNS. And that basically is for the unmet need for neuronopathic MPS II. As we know, the standard of care is enzyme replacement therapy, which does not address the CNS manifestations of the disease. So delivering a gene therapy that expresses a functional gene that can potentially have the potential to do have a functional protein. This is a one-time treatment to the CNS, and currently with the newborn screening, MPS II provides an opportunity for us to treat and identify MPS II patients early on and be able to treat them before CNS manifestations. So RGX-121, also now known as clamidogene A and parvovac, is the investigational gene therapy for REGENXBIO, specifically for the treatment of neuronopathic MPS II. So CAMPSITE was the study that was done in two parts. So part one was a dose finding study and part two was a pivotal trial. And I'm going to tell you a little bit about these. In part one, we enrolled 15 participants with neuronopathic MPS II. Ages were between 4 months and 5 years of age. Participants could be on ERT or could be naive. In part one, we did a dose finding, which means that we tried 3 different doses and 3 different cohorts or groups of patients. Immune suppression was given for about 48 weeks, and then there was an option to stop ERT at 1 year based on PI's discretion. Oops, sorry. The primary endpoint for the study was safety, and we had some other measures that we looked at to see if the product was working. These included heparin sulfate gags, neurodevelopmental assessments using the Bailey, caregiver-reported outcomes using Vineland, some systemic biomarkers, as well as hearing status in these kids. So part one, RGX-121, was tolerated across all 3 dose levels. There were no serious adverse events. So as we all know, when you don't have the I2S enzyme in MPS II, you have a buildup of GAG. And that's really the main insult that's causing the pathogenesis of the disease. So one common way and an early way to look if the treatment is working is to look at clearance of the heparin sulfate in the tissues and the cells that are causing the pathology in these patients. So for us specifically, and for severe or neuronopathic MPS II, we focus on heparin sulfate because that has high implications for brain and neurodevelopment. And for our program development, we specifically focus on a portion of that heparin sulfate known as CSF HSD2S6. And the reason we do that is because if you look at the bar graphs there, you see that that marker can differentiate your severe from your attenuated patient. So it is aligns with disease pathogenicity. The line graphs on the right, these are medians of the concentrations of HS-CSF D2S6 that are seen with the 3 different groups that we used, reflecting the 3 different doses that we used. And we see a very early reduction in CSF HS-D2S6 that is also maintained up to 2 years on the study. We have 1 patient in cohort 1, which shows a rebound. This patient had a shunt infection that was unrelated to study drug. So overall, we see up to about 85% reductions in CSF HSD2S6. And at week 16, which is an early time point we're looking at for the pivotal trial, we see about an 80% reduction in CSF HSD2S6. Dose level 3, patients were able to get to normal levels of heparin sulfate, and this was the dose that was taken forward to the pivotal trial. Some of the assessments that we look at to see if this drug is actually working in these kids are the neurodevelopmental assessments. And over here, I'm showing 3 subtests from the Bailey: cognition, expressive communication, as well as fine motor. I wanted to point out the gray shaded areas. These gray shaded areas tells you in a typical kid where the neurodevelopment should fall in a typical kid. And it's really important to consider disease baseline in these kiddos when they start on the treatment. So what I'm showing here are the participants that at baseline are they fall within that gray shaded area. So they are still within that typical development of these kids. And post-treatment, we are seeing that those patients are able to continue to acquire skills that keeps them within that gray area, which means that they are developing within the right trajectory. In this slide, I'm showing those patients that at baseline have more CNS disease impairment. So if you can see that they fall outside of that gray area. And our hope for these kiddos is that we continue to keep them stable and to prevent them from further decline. And what we're seeing here again are the same subtests, and we see stability and some improvements in some of these patients. The bar graphs that are colored, the greens show improvement compared to baseline, yellow is stability, and red is decline. And you can see across the 3 different measures that the majority of the participants are improving in each of those subdomains. We also looked at the Vineland Adaptive Behavior Scale, and again, the same color scheme here with green showing you improvement from baseline, yellow stability, and red decline. And you see the majority of the participants demonstrated stabilization of skill acquisition across the 3 different subdomains. And when we look at the Maladaptive Behavior Index, the majority of the participants that have an elevated Maladaptive Behavior Index at baseline show improved behavior, and those majority of them that have average behavior at baseline continue to be stable. So we know that hearing loss is progressive in these MPS II patients. So we looked at hearing. What I'm showing here is one of the common tests known as ABR. And if you look at the graphs on the left-hand side, you see two lines, an orange line that indicates hearing status at last time point, and the blue line indicates at baseline. And this tells you the hearing threshold and characterizes the hearing in these patients. And you see a shift between the two lines over time, indicating that we are seeing improvements in hearing. So we had data on 22 patients total. So we looked at each ear individually. So we had a total of 44 ears. And the table shows you that the majority of these patients, so 37 out of the 44 ears, have either improved or stable hearing at last time point. So one of the things we're looking at also is systemic effect of our product, despite the fact that it's administered into the brain. So we looked at continuation and discontinuation of ERT. And what we see with those level 2, we have about 71% of patients that are ERT free. So these are patients that were either naive and continued to be naive, or were on ERT and were taken off ERT. And with dose level 3, which is our pivotal dose, we see 80% of patients were able to be ERT free. So this points to the systemic activity of RGX-121. So I'm going to move forward and tell you a little bit more about our pivotal trial. So just as a reminder, this is dose level 3 that was taken forward. We had 10 patients that were enrolled, similar enrollment criteria. The primary endpoint here is focusing on the biomarker, which is CSF-HSD2S6. And our focus was to ensure that the levels of CSF-HSD2S6 reached to the maximum level seen in attenuated or below that. RGX-121 was tolerated in the pivotal trial. We had one serious adverse event that was related to elevated enzymes. This was transient and it was treated with steroids. So looking at our pivotal endpoint, which is the proportion of patients with the CSF HSD2S6 below the maximum level seen in attenuated patients at week 16. So we met our pivotal endpoint here, and you see in overall, we had 86% reduction in CSF at week 16. So we see this reduction in clearance of the substrate really early on. So just to summarize overall, RGX was tolerated across 25 patients enrolled in campsite. The pivotal dose reduced CSF HSD2S6 to levels that are below attenuated up to 2 years. The primary endpoint for the pivotal trial was met, and neurodevelopmental assessments demonstrate continued skill acquisition or stability in the majority of patients in dose finding study, which is part 1, and we continue to collect that data for our part 2 participants. We're excited to partner with Nippon Shinyaku, which is a company that has strong record in rare diseases, also in commercialization, to develop RGX-121 and our RGX-111 for MPS I, and to commercialize it in the United States and in Asia. So for RGX-121, which is for MPS II, we completed a biologic license application in the US, and we are seeking approval for the product. We expect FDA approval in the second half of 2025, so we're really excited, and we remain on track to be the first gene therapy and one-time treatment for the treatment of MPS II. For our MPS I program, RGX-111, we have restarted the development through our partnership with Nippon Shinyaku. We are excited about our interim positive data that we have collected so far, and we expect to pursue a pivotal program and an accelerated approval in the United States. Updates will be provided to Project Alive as information will become available. So I wanted to thank all the families that participated in our trial, and of course my colleagues, but I really want to thank you for being here and thank you for having us be part of this. We're excited to hear from you. We have a booth outside, come see us. And if you have any questions, please email us or come see us and we'll help. Thank you.
We are going to move right into our Q&A, and I will let you know that we did have one other presentation from Simon all the way in England. He was going to Zoom in, but that was unable to happen, but we do have one of his recordings, so if anyone's interested, it's on our website. If you don't know how to find it, just come see me and you can view that. So Q&A, I hope one of these works. Let's see, does that work? Is this working? Oh, yeah. Just wave your hand and I will come to you. I'm going to bring one over to you.
Hello. My question is it's in regards to the clinical trial you were just speaking about. So I know you said that typically there's an advisory board that will determine who gets into the trial, depending on the severity. How do you guys test or know, considering that there is a spectrum, how do you know which patients will have a more severe?
Thank you so much. That's a great question. So it is, I will say, also more relevant for certain conditions like Pompe disease, where there is an infantile onset form and a late onset form. How we determine, for instance, in that case, infantile versus late onset is actually by the gene mutations. And so a lot is known about what's called genotype phenotype correlation. So if we know the mutation, we know the symptoms and severity as well as the age of onset. And so that is the primary thing that we look at in genetics, but we also take into account the family history. As I mentioned, a lot of patients really are being referred to the trial because of a known family history. And for the most part, in a lot of lysosomal storage diseases, the ones at least that we're treating in here, there isn't a ton of variability in family members typically. I mean, when I say there's always variability in every individual, but when we're talking about, an infantile versus a late onset, there won't be that drastic variability. And so we take into account the timing of onset of disease as well as the severity in the sibling, for instance, and that will help guide us. As well as if we don't have a sibling, let's say someone comes because they're a carrier on carrier screen, we can use the literature again to look at, okay, is this mutation known to cause what we would consider to be severe or early onset disease? It is a bit subjective, the severity aspect, but I think what we basically just don't want to treat is a late onset Pompe disease patient who may never need enzyme replacement therapy until they're maybe 40 years old or something. That would be, in terms of a phase 1 safety trial, we want to make sure that the risk benefit ratio and risk benefit profile is really in the correct direction to not incur risk to the fetus and mother unnecessarily. Hopefully that answers the question.
Hi. I have a question about the clinical trials or the new, I guess, therapies that are coming out for hunters. How do you determine who is able to get into a trial if if I don't know the severity? Like my boy's mutation is so rare that there's no data on their severity. So how do I know if they're able to get into a clinical trial or able to receive the new gene therapies or new treatment?
Hi, I'm Bill Bucker from Denali. I'll take this one. Hopefully I can be a little bit helpful. I can talk somewhat specifically about what we're doing at Denali, but I think a little bit more generally to answer your question is that a lot of clinical trials, or pretty much all of the clinical trials that involve genetic disorders, there is usually a sort of a genetic advisory council that's created. I know in our trial for Denali, we have a genetic review committee that will sit down and look at all of the participants and all of the families who are interested, who can look really closely at the genetics. It does get a little challenging when the sort of phenotype, meaning the way that that gene is going to express itself in the patient. It can get a little murky if it is unknown, but generally in clinical trials, we do look it's looked at very carefully to make sure and sort of align everything up. I'm not a physician, so I shouldn't speak in these terms, but unfortunately, it's not an exact science. But there's a lot of things that are put in place for clinical trials to really get as deep as possible to evaluate the genetics and then also how they might translate to the patient. Does that help? And if there's any physicians in the room,
Yeah,
I realized halfway through my statement, I was like, I'm not a physician. So if there's any physicians,
I can explain a little bit more about, so even if it's what you call like a private mutation, meaning it's never been seen before in other families, but is sort of private to your family or a novel mutation. What we can still do as geneticists is there's different types of mutations, and these words might be familiar to some or others: nonsense, frame shift, missense. And so there's different types of variants, and they're sort of classified beyond just pathogenic or benign, which is disease causing or not disease causing. Within that, there are different categories of variants that can actually help us predict the severity. Because these are enzyme deficiencies or enzyme issues, there's also biomarkers that can help tell us, what is the level of enzyme, the endogenous enzyme that a person is making that they themselves can produce. If some of you know, carriers can actually have slightly lower enzyme levels than a typical person in the population, but they're not low enough to cause disease. But we can certainly use things like that, biomarkers like enzymes, other biomarkers. Or enzymes are not a biomarker. Enzymes is more definitive. Biomarkers, though, like glycosaminoglycans in the case of MPS, can also indicate severity. So there's lots of different ways that even if your mutation is completely private to your family, there are ways to predict severity. Again, it is not exact and perfect, but there are multiple different ways. Also, if an individual is already born, so that I'm talking about prenatal, right, when we really don't have a baby in front of us yet to examine. But if we do have a child in front of us, we can also do imaging and other studies that indicate, okay, how involved is the liver or the spleen? How involved are the eyes? And that all, collectively can help determine severity. As I mentioned, for the prenatal trial, we don't have phenotype. We're hoping we're getting treatment in before any symptoms actually occur. That's the goal of the trial. And so we rely more on biomarkers and genetics. But in these postnatal trials, we can use imaging and actual symptom presentation to determine inclusion or exclusion criteria. Does that? Yeah.
So just because I know you were kind of getting deep into that, our son was found on newborn screening, and I know what you're saying, but like the experts at UNC, it's actually in contradiction of what you're saying right now. Our son doesn't qualify for anything because he has a novel mutation, and you can also have something called a pseudo deficiency of enzyme. So he has zero enzyme activity, and even with that, you cannot use that as a biomarker, unfortunately. So we've been disqualified from everything, and it's just contradicting like what you're saying. So I just had to say that. I don't want families in here to have that type of hope. You know what I'm trying to say? Yeah, it's just right. But I'm saying, like you're saying, you could use biomarkers and everything to get someone in a trial, and that's just not correct. Okay.
I was answering the question of determining inclusion and exclusion criteria. Biomarkers is one of the ways in which we examine if they're affected or not. So enzyme is, like you said, can be falsely low due to a pseudodeficiency allele. But a biomarker like glycosaminoglycans, if that's elevated, that is a buildup of the metabolite. Correct. Yes. The genetic mutations in combination with biomarkers and enzyme and phenotype, all of that collectively is what I said, can help not put you into a trial, just determine what fits into an inclusion or exclusion criteria. By no means I'm saying I don't run all of these trials. I only run one. So I can only speak to the inclusion criteria for that one. I was just saying in general, when we're talking about gene therapy clinical trials, of which I run other ones for different diseases, we take into account all of these factors and then go, and sometimes they are excluded based on those results.
In our situation, our son, he would have had to have an older brother that was like proof of severe disease because he does not have a complete deletion, which would automatically say, okay, this will be severe. So I can't remember at this point because his whole first year is kind of a blur, but I think he has a missense or something that can go either way. So basically, the only way he would have gotten into a trial, even though we have newborn screening, is if he had a severe mutation that they already know, okay, this is a full deletion. that kind of thing. So that was just to my point, because I think a lot of people like clinically say things, but like what a parent hears is a lot different than what I'm trying to say. Because like, right, you kind of hold on to anything. So that's why I just wanted to share our experience, just because that is unfortunately a reality. And it's becoming more prevalent because of newborn screening. So it's a unique situation, unfortunately. Yeah. Yeah, I understand. It's just, yeah.
Yeah, I just wanted to follow up and say, to your point, and to what Bill said and what you said, each family is looked at independently, and a variety of factors are considered when we consider enrolling them for the clinical trial, including their enzyme levels, their biomarkers, their genotype, and each patient, because each patient is an individual in this situation, and we're dealing with a rare disease and with different genotypes, that we really have to consider each patient as an individual. And which is why it's so hard to answer your question with a simple, this is how we do it, because we look at your patient and we look at your child and all of those factors when we consider, can we enroll them in this study? And we really do want to enroll them in this study if we can.
And I wanted to add one more thing, and we also look in our child for neurocognitive testing. So again, it's a multiple things they could look at. So genetic determination, family history, but you can also do genetic testing early, and that could help determine survivors.
Just to add to that, though, there are unique things with that as well, because we also looked into developmental testing, but unfortunately, because again, this onset of degeneration and all of that is a range, right? Also, treatments they're on can affect that. So a lot of the time, trials, at least in the trial form, even with developmental, screenings, there's a lot of pushback with that as well because the onset could be later. So I'm just sharing parent perspective here. I actually do have a question though, if I can. And I don't really know exactly who this would be directed to. I imagine it would be the Takeda speaker, and I apologize, I forget his name. I just wanted to see, have you guys experienced any changes as far as the newborn screening advocacy you do based on the administration changes that happened? I know that that committee got disbanded. So I just kind of wanted to see, especially because, our family was very blessed to have newborn screening, and we actually got caught on a pilot study in North Carolina, kind of as a fluke. Like it's technically not even on the newborn screening here. I just wanted to see kind of what, if anything, if you've heard anything, or has that affected your support in that way?
No, I think it's a question that everybody, of course, is trying to grapple with. These things change almost on a daily basis. So we're trying to work with advocacy partners, with folks outside, with the policy groups as well, to figure out where not just Takeda but in general all the other companies that are operating in this space need to respond to. Uh, from strictly from Takeda's perspective, what we have decided to do is, after we work with the advocacy groups, get MPS2 on the newborn screening panel, we have now shifted our commitment towards helping actually now implement newborn screening. So one of the things I mentioned earlier was that we are working with the experts as well as the advocacy partners on a newborn screening practical recommendations publications, because there are a lot of sites that really don't know today what to do with newborn screening if they have a positive case. And that's a huge problem. So as new modalities will come to market, we realize that this is going to be a challenge. So we are continuing to do that. And of course, it's very refreshing that for a change in a very, very long time, we have more partners in this effort. So we are looking forward to seeing how that all evolves, and we'll continue to support the best we can.
One thing too, I mean, obviously, a lot of changes happen that is, you know, in terms of leadership and different entities that we in the rare disease community have worked with over time. But the one good thing when it comes to that committee being terminated, newborn screening is not terminated. It's still a state by state program. And so, you know, we are still going to keep moving forward with advocating it with all the states to add Hunter syndrome to their newborn screening panels. And we're just going to, as we're used to in our daily lives of having a child with Hunter syndrome, just keep moving forward, you know, and adapting and figuring out what will the new path be. The path hasn't closed, it's just changed. And so we're going to have to kind of watch and see and figure that out. But as Ravi mentioned, we have really great partners in Every Life Foundation and NORD who really lead the way in the rare disease policy world. And we make sure to really stay informed and partner with them to make some of those changes. So we know how important newborn screening is, and we're going to keep moving forward with that. And that's something too that if anybody is passionate about that, we need your voice. If it's not currently in your state, the only people that they'll really listen to are families like ours who live in their states. So we need your help in making that happen. I think we have a couple more minutes. Okay.
I don't know how to say this right. So from what I gather, there are two treatments that are hopefully going to be approved by the FDA. Technology and gene therapy. So I know with the clinical trials, if you're in one trial, you have to wait a certain length of time for participating in the whole trial. Once they're both approved, and I'm sure it's probably like anyone's guess, because we don't know for sure, and I'm not going to hold anyone to anything, but is there any idea of how it would work once both are approved? If you're in one trial currently, you just jump right into the something that's approved? Or is there still going to be a waiting period? You have to wait. Like, in other words, do you have to show a decline, go back to some kind of a decline before starting something? So let me make sure that I understand you correctly. So you're saying if you're already in a trial, currently in a trial, and then there's something approved, yeah, there's going to be treatments approved. Right. So the trials will end once the approval happens. So if you're in one trial now and then everything gets approved, now you want to start the other, what would that look like? So if you're already on the trial and that same product gets approved, you'll continue to be monitored as part of the trial, right? If you're on a trial, but now you want to try a different product, it's going to depend on what type of trial you are on. If it was a gene therapy trial, for example, it may not be possible for you to get another gene therapy. If depending on also the label that comes out specifically for that trial and what's the eligibility criteria. So there's going to be a set of eligibility criteria that are going to be similar for you to be able to take that in. We don't know right now, but those will be broadly available as part of the label for the product or for the drug. And then that will be also another consideration, I guess, as payers, right? So I'm not really very familiar with that aspect, but also related to sort of how the payers would, you know, whether they would allow or not allow, so.
Yeah, in general too, I think one of the things to remember is that there are certain things that really the FDA is in control of, and kind of determining and approving a specific label once a therapy is approved is in their court. That ball's in their court. So there are some unknowns still. And then ultimately it does vary, as we've seen, even in terms of the clinical trial criteria itself, on what you can and can't do, and kind of go from one thing to the other, which will be more clear post approval. And then ultimately, it's always going to be up to your own doctor, right? That once a drug is approved, it would be a prescription. So that's something too that you would be consulting with your own clinician around, and then being able to determine the best course of action. And hopefully this time next year, we will have a whole different topic on how do I choose between which therapies? I mean, what a world. So just want to take a moment to think about that, how huge that is. I've told many of you before, you know, when my little brother was diagnosed in 1998, there was nothing that they could do. There was no treatment whatsoever. Some of you here may remember that. And, looking at the world today when babies are born, it's first of all, you can get diagnosed as an infant and you can receive treatment, and hopefully we will have treatment options that you're getting to choose. And there is a burden of choice, and it will bring along some new challenges that we will face, but what a good problem to have. So it's a really amazing time to be part of this community and to see it. It still gives me chills, and I still can't believe we're here. And like I've said earlier this morning, yeah, next year, I hope this is a party rather than a sit down conference. Okay, we'll be up on our feet. So, all right. If you all have any more questions, everyone has a